folks, we are going to begin chapter 10. Actually, let's see. I'm going to shrink myself just a teensy bit more. There we go. Okay. And here we go. Okay. So the main things that you need to know are the cell cycle and what happens within each phase, mitosis, and what happens within each phase of mitosis, and the differences between mitosis in plants and animal cells. So the overall function of cell division is making new cells for either reproduction, growth, or repair and renewal. Okay, so main functions of cell division: growth, repair, renew, reproduce. When we talk about the cell cycle and we talk about mitosis and meiosis, okay, we're going to use some terminology, and we refer to, when two cells divide, we refer to the cells, the new cells, as daughter cells. Now, this doesn't actually imply any gender or sex to the cells themselves. It's just a term that we use. We call the cells they came from parent cells. So just be familiar with that terminology. So you need to know what happens in each phase of the cell cycle. So interphase is 90% of a cell's life doing its cell job. So whatever the cell's job is, whether it's a nerve cell, a bone cell, or a muscle cell, it's doing its job, and it's growing in size. There are 3 different phases. There's G1, S, and G2. Okay, so you need to know these phases and what happens during them. Okay, so G1, we call this the first gap. We call these gap phases. And this is the cell doing its job and growing. So the first gap phase is mostly growth, and this is also where we receive the signal to divide. S phase is the DNA replication or the synthesis phase, and this is where we're going to make copies of our chromosomes. And then G2 is the second gap phase, and this is where we're going to prepare for division, where we're going to produce different pieces of equipment, like parts of the cytoskeleton, like centrioles and microtubules, that are going to aid in cell division. And then we enter mitosis itself, which again is part of the cell cycle, but it's divided up into 4 phases: prophase, metaphase, anaphase, and telophase, or PMAT. So when we are going through our S phase or our synthesis phase, we are making copies of our DNA, and we need to make identical copies because when we are going through mitosis, we are making copies of cells, identical copies, like clones. Okay, and this is actually where we get mutations, is during the synthesis phase, and that's important to know because mutations can lead to genetic variation, or mutations can be bad. So when we divide cells, we are dividing both genetic material and the cell and the parts of the cell itself. So we have mitosis, and then we have cytokinesis. And cyto means cell, kinesis means to move. So this is literally cell movement, and this is the actual splitting of the cells into 2 new cells. So this is what's called a karyotype. And we have 22 pairs of autosomes, or body chromosomes, and then 1 pair of sex chromosomes: XX, XY, XYY, YY, XXY, just X, just Y. It's a spectrum. We'll talk about it more. So 23 pairs total, 46 chromosomes all over. Okay, and this means we're diploid. Meaning we have 2 sets of chromosomes. Why do we have 2 sets of chromosomes? Because we get 1 chromosome from mom and 1 chromosome from dad, and they're identical. Okay, so the chromosomes like chromosome 1, pictured there. Okay, you can see little bands on it, and those bands are genes. And those genes code for the same traits. We're going to talk about this more in the next chapters. And this is where we get some of our genetic variation that we'll talk about in a minute. But these are what we call homologous chromosomes because, remember, homo means same. So they are, because they have the genes that code for the same traits are located in the same place. Okay. The only cells in your body that are haploid, meaning they only have one set of chromosomes or 23 chromosomes total, are your sex cells or your gametes. Why? Well, when we sexually reproduce, we bring together egg and sperm, which are your sex cells or your gametes. And you wouldn't want them to have a full complement of chromosomes each because then you have too many chromosomes. So you have your X and your Y, and they each have, or normative X and Y, and they each have 23 chromosomes each. So when they come together, you have 46 total or 23 pairs. So we use parts of the cytoskeleton to help move things along literally in mitosis. So we use microfilaments, microtubules, centrioles / centrosomes. Okay, so centrioles and centrosomes are kind of used interchangeably, and as are sort of like microtubules and spindle fibers. So you just kind of need to know when these show up in the processes. Okay, plants don't have this stuff. So this is the difference between plants and animals. Is animals have all this structural apparatus to help with mitosis, whereas plants don't. Okay, so what happens with chromosomes? So when your DNA is in your cell, in the nucleus, it's just hanging out. It's just chilling. It's relaxed. It's unwound. Okay, then it's going to get wound up into and around these little histone proteins that are then going to form these little solenoid coils that are then going to turn into these chromatin loops, which are then going to kind of turn into these little rosettes around a scaffolded protein, and eventually they will condense to become a mitotic chromosome, which is sort of what like we picture a regular chromosome to be. Just you need to know that DNA is wrapped around histone proteins, which becomes loops of chromatin, which condenses into a mitotic chromosome. So in G2, okay, what happens? Chromatin starts to condense. All right, so that's where we have that DNA being wrapped around these histone proteins, okay, and it helps to keep the DNA organized during division. So before replication, homologous chromosomes, remember, are just like one little chromosome by itself, just one little bugger, okay? And then they have these little kinetochore sticky protein buttons on them, okay? And they have the center spot on them called the centromere. Kind of makes sense in the center. And when these chromosomes are duplicated, okay, after replication, after S phase, okay, this is what they look like. They look like these homologous chromosomes, where now, instead of just like you have the blue one that's from dad, the red one that's from mom, now you have the sister chromatids, which means you have 2 from dad, 2 from mom, and they're connected by their kinetochore sticky button proteins at their centromere. Okay, so you need to know that anatomy. You need to know the anatomy of a homologous chromosome, that they're referred to as sister chromatids, and they're homologous chromosomes in this form, and they have these little kinetochore sticky proteins that stick them together at the centromere to make them into the sister chromatids / homologous chromosomes. Okay, so we are going to go over We're going to focus more on the 4 main phases. We're not going to focus as much on pro-metaphase. Okay? So we're going to go, you need to know the different steps of what happens in each phase of mitosis. So in prophase, okay, we have our centrioles moving to the opposite poles of the cell. We have our DNA condensing. Our nucleolus disappears, goes away. And our nuclear envelope, okay, the membrane around the nucleus that houses the DNA breaks down. This allows us to have access to the DNA to be able to split the DNA and go into 2 cells. We talked about that. Okay, metaphase. Meta means middle. Sorry, pro means first, so prophase comes first. Meta means middle. Okay, so that means the chromosomes are going to line up in the middle or on the what we call the metaphase plate, which is just the middle of the cell at that time. Okay, and that's the main thing you need to know about this, is that in metaphase, the chromosomes are aligned along the metaphase plate. Okay, so let's a look at that. Now, in anaphase, okay, our sister chromatids are going to separate. They're going to be pulled apart. Ana means up. So our chromosomes are going to move up to the poles of the cell, okay? And they're pulled at those centromeres, that little area in the middle, okay? So Ana, up. The chromosomes, the sister chromatids are pulled apart into individual chromosomes up to the poles of the cell. Telophase. Telo means last, so this is our last phase of mitosis. Okay, and this is where basically most of the events of prophase are reversed. So the chromosomes are now at the opposite poles, and they start to unwind, because remember they condensed before, so now they're going to start to unwind. Our nucleolus is going to come back. Our nuclear envelope is going to reform, and our spindle fibers are going to go away. Okay, so chromosomes, the DNA begins to unwind. Our nucleolus comes back. Our nuclear membrane breaks down. Our spindle fibers go away. And we have the beginning of actual cytokinesis. So it's sort of like telophase / cytokinesis, because cytokinesis, which is again the actual splitting of the cells, starts during telophase. Okay. How this happens is through this little muscle ring belt called a cleavage furrow that forms between the 2 cells, and it kind of just like tightens up like a drawstring and then splits the 2 cells. Now this is somewhere where animals and plants differ, where animals have the cleavage furrow, okay? Plants, they have a cell plate, okay? And the cell plate actually becomes part of the new cell wall, all right? So animals, cleavage furrow, muscle, like ring of muscle that just like tightens off and snips off the cells. Plants, they have a cell plate that eventually becomes the new cell wall. Okay, so remember again, for each stage, well, you don't need to know this for each stage, but for prophase and telophase, what is the status of the nuclear envelope and the nucleolus? What is the structure of the chromosomes? What's happening with our centrosomes and microtubules? What are the differences between plants and animals? Okay, plants don't have all of the centrosomes and microtubules and structural elements. Okay, and they have a cell plate instead of a cleavage furrow. Okay, here's some review. So a little bit about cancer. Okay, so cancer is uncontrolled cell division, which means certain checkpoints of the cell cycle failed. So there's a special protein called P53. Okay, and when that protein maybe gets a mutation or something's wrong with it, it allows damaged DNA to get through, and then that damaged DNA can begin to divide uncontrollably. And then this is when we have transformation, which is when a normal cell is converted to a cancerous cell. Then we have the formation of a tumor, which is when we have a bunch of uncontrolled cells dividing in one area. A benign tumor is at one site, it's not spreading, whereas a malignant tumor means that cells have spread, the cancerous cells have invaded or metastasized into other tissues to form additional tumors. Now, we usually treat this with chemotherapy and radiation, though immunotherapy is having a lot of really good results with a lot less negative side effects on the body. Okay. So, I will see you in chapter 11.